I'm alive. Good. It's good to see all of you again, even though I can't see you. But I am glad to be back online. I would have gone earlier today. I would have gone online earlier, but we had to have a video conference with one of the new deans, and he informed all of us that everything's going to be online for the rest of the semester, and everything's going to be online for the summer, and more than likely for the fall as well. And they will have hybrid courses. Those are the courses that have to have labs attached to them, and they may be able to open up the campus for a hybrid course, but they're still working on that. They're not quite sure how that's going to work. So we just have to wait and see. But things will be online all summer, and it will be online in the fall. Hopefully by the springtime, things will settle down and we'll have classes again face-to-face. Okay, we are going to discuss a little bit today about taste. You did read, and we discussed about the sense of smell or olfaction. We're going to talk about the gustatory sense today, which is our sense of taste. And if you look on lecture 13, and then in your textbook on page 456, it will show you a diagram of the tongue and an illustration, an artist's illustration of what the papilla look like on the surface of the tongue. Those are those structures you can see. If you look at your tongue in the mirror and you rake your teeth over your tongue, sometimes they look to be whitish in color and stand up. Those are papilla, and your sense of taste is gustatory. That sense of taste is chemical, depending upon the chemicals in your food, and your ability to taste is registered as sweet, sour, salty. There's also fat involved in that, and bitter, which is the worst. But those are the senses of taste by the papilla, and the gustatory sense is what registers. Those are sensory afferent neurons that send the message to the brain as to what you are tasting. That sense of taste comes to us because of the facial nerve, the glossopharyngeal. Remember, the glosso is tongue, pharyngeal is throat. Tongue does have a facial nerve for that. So that those are two structures. And then the vagus nerve, which was the 10th cranial nerve, also picks up on the sense of taste. After the sense of taste is vision. And your textbook has devoted a great deal of information on vision. And if you look in your textbook, it begins on page 469, where it says sense of sight on 469, and it's extended to page 484. Your reading assignment will be 469 to 484. I also would like you to watch 2 videos for me. And the reason I want you to is because it will be much easier for you to see these structures with their cameras set up. All I have is this little camera here, and I guess that's what we're going to be looking at if I show you the diagram of the eye. It looks better at that angle, doesn't it? But we'll look at your text diagram as I talk about these structures. But if you look at these 2 little videos, part 1 and part 2, it's excellent. And you can see all of these structures pointed out. They describe how they're built and their physiology. It is eye structure and function part 1 and part 2. I also want you to do a virtual lab, and all you have to do is look on YouTube under the little eyeglass, Professor Wilson, sheep eye dissection. And he does a very good job of that. And you'll understand that dissection much better after we finish or conclude our discussion of the eye today. Now, vision. Vision is a sense that is carried to the brain by the optic nerve. The optic nerve is a large cranial nerve. It's the second cranial nerve coming off the brain, along with the olfactory nerve, which was number one. Vision is number two. The olfactory is the sense of smell, and vision is optic. The environmental stimulus for vision is light. And the receptor organ for vision, of course, is the eye. The eye is a structure that forms embryonically, and it is an outpocketing of the brain. And so when you look at someone's big brown eyes or big blue eyes, what you're actually looking at is nervous tissue. Everything else you see is either dermis, which is skin, or a product of the dermis, which is hair. But when you look at the eyes, they're an outgrowth of the brain. They're heavily protected because they're set back in this bony orbit. And remember, we studied that bony orbit in detail when we talked about the skeletal system. And we also learned that some people have a higher process at the top of that bony orbit, and that is the now it just eluded me, supraorbital ridge. The supraorbital ridge, all of that is highly protective of the eye. The eye is a vital organ, and in some respects it's sturdy, in other respects it is delicate. So you have an eye protected by the bony orbit, as well as the eyelids and the eyelashes above on the upper lid and the lower lid, which catches dust and pollen, bacteria. You also produce tears, which are very water-like in consistency, but they do have a specific gravity. They do have, in those tears, salt, which you don't find in distilled water. You have salty tears, which is an antibacterial agent, and you also have an antibacterial agent by the name of lysosome, Lysozyme. And that is in your notes. Okay. Another structure that protects the eye, and you've heard probably of this, is the conjunctiva. The conjunctiva is a thin, transparent mucous membrane that tucks in under the lower lid, goes over the eye, and then tucks in under the upper lid. We're aware of it because when people get an eye infection, more often than not, it is conjunctivitis, which means inflammation of the conjunctiva. Conjunctivitis can be caused by a bacteria. Consequently, it's bacterial conjunctivirus. And another conjunctivitis is viral, caused by a virus. So that conjunctiva can be inflamed when it's infected and produce a great deal of mucus to try and get rid of that infection, as well as a lot of tears, which, as we've told, has an antibiotic agent. But depending upon whether it is a viral or whether it is a bacterial infection will depend upon what topical antibiotic is used. And sometimes it's a liquid antibiotic, and sometimes it's a salve or a cream-like structure that you'll put in the eye to overcome that conjunctivitis. When a child gets conjunctivitis in school, they immediately call the parents. The parents have to come pick up that child because viral or bacterial conjunctivitis, inflammation of that conjunctiva, is extremely contagious. Now, if you look at the eyeball itself, as illustrated on page 473, there's a mid-sagittal section through the eye. You can see it right here, and figure 12.25 on page 473. I'll point to that structure if I can on my transparency. And as I discuss the structures, I want you to note those structures on that page as I describe the structure and function. Okay. And the first thing that we want to mention is that the eye itself has three tunics or three coats, three protective coats. Just like the brain has the meninges, the dura mater, the arachnoid, and the pia mater, the eye also has three. And the outer eye, or the white portion of your eye, which is what you can see on the outside of your iris, that is referred to as the sclera, S-C-L-E-R-A. It is not labeled here, but I chose this because it's a big eye. This would be the sclera. It's the white portion of the eye. It's extremely strong, very durable, and sadly, we can't dissect the sheep eye. You will see it when you view Professor Wilson's sheep eye dissection. And the only thing I can say is that you'll be amazed at how sturdy that thick, heavy sclera is. And when you pronounce the word sclera, the S sounds and the C, S-C-L-E-R-A. It is thick and it's sturdy. It would be difficult to puncture the eye, but it has been done. And if the eye is punctured, the fluids in the inner eye, in the back portion of the eye, the posterior eye, that's the vitreous humor, can ooze out. And if that does occur, then the eye has to be enucleated, or blindness results from that. But that's the sclera, the thick, heavy sclera. The next layer, which this artist has done in blue, and if you were actually able to look at it, it would be a very, very dark color. That middle color coat, that middle coat is called the choroid coat, C-H-O-R-O-I-D. Remember the choroid plexus. The choroid plexus was that place in the pia mater that is highly vascularized with thousands of tiny capillaries that extract from the plasma, the cerebrospinal fluid. Well, choroid plexus, the word choroid means it's highly inundated with blood vessels. And that choroid coat, which is the middle coat, is full of thousands of tiny capillaries and pigments. If you look at it, it appears almost black to the eye. It is just a dark, dark, dark color. And the reason for that is that the eye is very much like a camera. And when light comes through the eye, the light is bent by the various refractive media of the eye, so it falls right here in a little area of acute vision. And we'll talk about that, the fovea centralis. But that light is bent, and so that it falls right on that fovea centralis, which is up above the optic nerve. Now, if we didn't have that heavily inundated choroid coat, if it was not real dark, then light rays could reflect in different directions and interfere with vision. So that's like a camera. The inside, that choroid coat, is very, very rich in blood vessels and heavily pigmented with melanin. Now, right in front of that is the retina. And the retina is the most important layer, if you can have an important layer, because they all are, but the retina is significant because it contains all the photoreceptor cells. The photoreceptor cells we'll name as the rods and the cones that pick up on light and color. And it is rich in the retina, in those photoreceptor cells. But that retina has to be fed. and it is fed on the choroid coat right behind it. So all those capillaries inundating that choroid coat, uh, which extract from the bloodstream all those rich vitamins and minerals, enzymes, they all feed that retina in front. So those photoreceptor cells, the rods and the cones, are fed by that choroid coat. Then on top of that is the retina. Now the retina, if you look at the retina in the sheep eye, and hopefully you'll be able to see that in Dr. Wilson's dissection of the sheep eye, that retina looks kind of like tan nylons. It's a beige color, and you can actually pull that retina. You can tug on it a little bit and pull it away from that choroid coat, and then you can really see how black that choroid coat is. If anything gets between, for example, if any fluid leaks in between the retina and the choroid coat, separating the retina from that choroid coat, those rods in that area and cones can begin to die because they're not getting the oxygen in the food. They're pulled away from the choroid and they're not being fed. So that is what they call, it's a dislocated or a retina that has detached. And you may have heard it as a detached retina. When that occurs, then it has to be remedied pretty quickly with laser surgery, because if it goes for very long, vision can be lost in that area. So any heavy blow to the head, football players have gotten quite a few of them, nothing like boxers have, who get pummeled in the head quite a bit and detach that retina, and some of them have gone blind from it. So another reason you don't want to get a good knock on the head. But those are the three: the sclera, the choroid, and the retina. They're called tunics. Remember, everything was named, came to us from the Romans and the Greeks, and the Romans wore their clothes in tunic fashion around their body. So that was a layer of cloth. That's why they referred to these three layers from the outside going in as tunics: the sclera, the choroid coat, and the retina. If you look in your notes, lecture 13 on page 32 of Neuro, you'll see a drawing of a rod. It kind of looks like a squid. It really does. And you look at it, and you can look at it here. This is what I'm talking about. You can see the nucleus in the cell body. This that looks like a proboscis or a nose is a dendrite. There's the axon, and these are collateral branches going off the, the axon, which will release neurotransmitters to the dendrites of the next sensory neuron, carrying the message to the brain via the optic nerve. Now, the rod itself works in dim light. If you've ever gone into a theater and the lights are down low, they're dimmed, and it's noon and you're going in, and the lights are dimmed, you can have difficulty. You can see, well, I can't even see any aisles, or I can't see the chairs, and you have to stand there for a few minutes. Well, in those few minutes, in that depressed amount of light, then there is a pigment that fills up this dendrite, and the pigment is referred to as rhodopsin, rhodopsin. It's also called visual purple. And once it fills up all the way and goes to the end like this of the dendrite, then you can start to see. It takes a few minutes. So the next time that happens to you, just realize that that dendrite has to fill up with that pigment before it is able to be stimulated. And the rods are important for picking up objects in dim light. What happens is the light that is provided bounces off those objects to your eye, and then the refractive media of your eye, your cornea, your aqueous humor, through the pupil, through the vitreous body, and then it falls on the fovea centralis. It's not registered. It's not registered until those rods fill up with the rhodopsin, and then they can pick up the light that's being reflected off those objects and then bent through your eye. But those are the cones. The cones are here is the fovea centralis, which is the little dent that's the area of most acute vision. And if you look just above that, in this area, kind of in the peripheral area, there you have your concentration of rods. And then in the little depression here, you'll have that little depression in the retina, which is known as the macula lutea, that little depression, and it's filled up with cones. That has your highest concentration of cones, and of course that is where your most acute vision is. Those cones, and you'll see the cone kind of looks like an ice cream cone below the rods. These are the cones. It has a shorter dendrite and a fatter dendrite. Here, there's the cell body, the axon, the collateral branches. The cones are responsible for picking up on color. And the three colors that register, red, blue, and green on those. Those are the primary colors. Now, the cones pick up on color because they also have to have a pigment which fills up those cones, that is in those cones, and that pigment is iodopsin, and that is on page 33 at the very top. These rods and cones, they are the photoreceptor neurons, the afferent sensory photoreceptor neurons contained within the receptor known as your eye. The rods and the cones are the photoreceptors. They're in the retina and layered in the retina, and the retina lies right against, again, the choroid coat full of blood vessels that feed that retina all the nourishment it needs. One vitamin that is extremely important, not only for the health of the conjunctiva and the manufacture of tears, but also the health of these pigments, especially rhodopsin in the rods, is vitamin A. Vitamin A can be found in liver. It can be found in meat and yellow and orange vegetables, pumpkins, squash, carrots, all of that, high in vitamin A. And that vitamin A helps protect those rods with an ample amount of rhodopsin, and also protects the conjunctiva so that it stays moist, and also that the lacrimal glands up on the upper lid, they can secrete the tears over the front of the eyes in ample amounts. Now, if you look below the discussion, there is a discussion on color blindness, and of course it occurs when one or more of these cones are defective, and the defective cones occur. That's a hereditary male pattern baldness, as well as color blindness and hemophilia. All of these are sex-linked chromosomes on the X and Y pair, the 23rd pair of chromosomes, and that failure to see and distinguish between red and green and sometimes blue, that is what we refer to as color blindness. The individual that's completely colorblind and sees nothing except in blacks and grays, that is complete color blindness, and it does occur, but it's extremely rare. Now, I did mention to you that light has to pass through the media, all the layers of the eye, so that the light can fall directly on the fovea centralis at the back or dorsal side of the eye above the optic nerve. Now, we'll go through and I'll talk about these structures and I'll name them first and then I'll go back and talk about their physiology. The first structure that light has to pass through, sometimes they call it the clear window of the eye. That first structure is the cornea. It's a convex structure. That means it bulges out. That is the cornea. Over that cornea, and the artist has rendered this, there's the conjunctiva that is over that cornea. And remember, it tucks in underneath the lid. But that is the cornea. And the cornea may look delicate, but it is pretty sturdy. It is actually a downward projection of the sclera, and it's a transparent downward projection of that sclera. Right behind the cornea is a what they call the anterior chamber. That is a little area behind the cornea and in front of the lens. And this anterior chamber is filled with fluid. That fluid is known as the aqueous humor. Aqueous humor. Aqueous blue humor fluid. Then it appears to be a hole in the eye, and it's just an opening in between the iris. The iris is a circular muscle that is around that hole, the hole we call the pupil, and the iris controls the size of the pupil, and we'll talk about that soon. So light goes through the cornea, then through the aqueous humor, it passes through the pupil, then it passes through the this biconvex disc known as the crystalline lens. Biconvex disc. Now, that disc is held in place by ligaments right here. And you can see those ligaments diagrammed on page 473 as well. These little ligaments are suspensory ligaments, and they're anchored to an envelope that the crystalline lens is within, is inside. On one side, the suspensory ligaments hold on to the envelope containing the lens, and on the other side is what they refer to as a ciliary body. Sometimes they call it ciliary muscle, Ciliary, but I like to use the word body because it is both smooth muscle and it also has glandular tissue in it as well. Smooth muscle and gland. That's the ciliary body. Now, behind the lens is an area, a large area, referred to as the posterior chamber. And the posterior chamber is filled with a transparent, thick, gelatinous substance known as the vitreous body. Sometimes they'll call it the vitreous humor. I like the vitreous body a little bit better because it tells you about how substantial it is. That's the vitreous body. Now, the light falls all the way to the back of the eye, above where the optic nerve is. This is the optic nerve that is carrying all of those sensory afferent neurons, rods and cones. They exit right here at the optic nerve. And that optic nerve just in front of it is what they call the optic disc. And the common name for that is the blind spot. They call it the blind spot because that area does not have rods and cones. So optic disc is the scientific term for that, but optic disc, but the other term for that is the blind spot. And then above that is the little depression. Now, the little depression, this depression occurs down into the retina and a little bit into the choroid coat. It's kind of like if you took a spoon or an ice cream holder and you dipped it into ice cream and pull the ice cream out. There's a depression in the spoon that has a little depression in it. The depression itself is called the macula lutea. Sounds like a opera, an Italian opera. Macula lutea. And the macula lutea is filled with cones. And because it's so heavily inundated with cones, it's considered the area of best acuity or best vision in that area. And because of that, it's dubbed the fovea centralis, the central area with the highest acuity for vision, right there. The depression is the macula lutea. The fovea centralis is the name of that structure, or those that little bulge that lies right down in the macula lutea, filled with cones. Now, you also have muscles, and you have ribbon-like muscles that insert on the sclera. You have a muscle above, and these muscles are attached back behind the optic nerve by what we call the annular ring, and that is a flat muscle above and below. When this contracts, then the other muscles relax, and you can elevate the eyes in vision and looking up. When it relaxes and this one contracts, you can look down. It doesn't show it here, but you've got flat ribbon-like muscles that are also lateral on both sides of the eye that enable you to look to the left and then to the right and then to the center, as it occurs when you cross your eyes. Okay. So that is just the names of the structures that we've been talking about, just the names of the structures. And now I want to talk a little bit about their physiology. We'll start out with the cornea. Now I want to keep in mind that the entire eye is heavily inundated with millions of afferent sensory neurons for touch, pressure, and pain. If you get anything in your eye, an eyelash or dust or sand, it feels like a 2x4 until you get it out or wash it out. And the reason your eye is so terribly sensitive to that is because you have all these wonderful afferent sensory neurons that pick up on the presence of a foreign object in the eye. And if you get anything on the cornea, it's also very, very sensitive. As I mentioned, the cornea is a forward projection of the sclera. It is the clear window through the eye. It is convex in nature. It bulges out. And what's important is when light passes through the cornea, it is bent, as you can see by the artist illustrating the line here. It is bent so that it will pass through the aqueous humor and the pupil. Most of the refraction, and when we talk about refraction, we're talking about bending of light. Most of the bending of light, most of it occurs right here because of the cornea. And for that to be, to occur in a healthy manner and in a positive manner and an effective manner, that cornea has to have a very rounded, convex curve to it. Sometimes the cornea is irregular. It almost has kind of a wave to it. And when this occurs, then the refraction is not accurate. It'll bend light in a way that is not conducive to good eyesight. When there is an irregular surface on that cornea, that is referred to as an astigmatism. And it can be corrected by your glasses or your contacts, and LASIK, which is how they'll go in and they can reshape that cornea. They can reshape the cornea in a manner to avoid and reshape it so that it eliminates that astigmatism. The same thing can happen in LASIK if they want to correct for near or far vision. They can reconstruct that cornea so that the light refracts or passes through the cornea in such a manner that it'll effectively reach the fovea centralis. But that's the cornea, clear forward projection. Now, it doesn't have any blood vessels in it. It is fed by the fluid that is behind this cornea. And the fluid that fills this anterior chamber is the aqueous humor. The aqueous humor is to the cornea as cerebral spinal fluid is to the brain. It's highly nutritive and feeds those corneal cells, keeps them nice and healthy because there's no capillaries to feed those corneal cells. Now, where does that aqueous humor come from? I mentioned the ciliary body, but I want to mention again that this ciliary body, sometimes called the ciliary muscle, it's better named body because it is smooth muscle as well as glandular tissue. And the glandular tissue in the ciliary body extracts from the plasma the contents of the aqueous humor. It extracts and sends it in to that aqueous humor. It will actually flow behind in this direction. It'll flow behind the iris and then come out through the pupil and bulges the cornea out, fills that anterior chamber with fluid, the fluid known as the aqueous humor. Now, that aqueous humor is constantly being made by the ciliary body, and it's constantly being reabsorbed. The reabsorption of that aqueous humor occurs through a canal. It's not labeled here, but let's see if it's labeled on this one. It is called the Canal of Schlemm, and lo and behold, it is not labeled there, but it is on this structure. You can see that. And that Canal of Schlemm is kind of a little suction at the bottom of the eye, and it pulls in constantly, reabsorbing this aqueous humor as it's being made by the vitreous body. Okay, I'm going to break a little bit here, and I promised you that we would have our next double quiz. And let's say I will ask you to be sure and get this double quiz in by tomorrow evening at 7 o'clock. It's a little after 5 right now, but tomorrow evening at 7 would be good. And that'll give you a little more than 24 hours. So I promised that I would ask you some questions on 11 and 12, as well as a few questions on the eye as we pass through its discussion. So if you number a little paper to the side, 1 through 10, I'll give you your first question. All right. If you remember in 11 and 12, we discussed the fact that in the thoracolumbar region of the spinal cord, there are, on either side of the spinal cord, there is a number of ganglion all the way down in the thoracolumbar region. And we call these ganglion, which are involved in the fight or flight response. We say they are lateral ganglia. We also call them paravertebral ganglia because they're right next to the vertebra. And there's a third synonym for these fight or flight ganglia. What is the third synonym for these lateral ganglia or the paravertebral ganglia? That was for number 1. Number 2, and all of you should get this one. All of you should get all of these because you can go back and listen to the tapes and get your answers or look in your notes. So all of you can make hundreds on these. And this is a double quiz, so it's double hundreds. On number 2, what part of the nervous system, in a diagram of the nervous system, what part of the peripheral nervous system is under unconscious control? What part of the peripheral nervous system do we not control? The heartbeat, the digestion of our food, even the eye muscles that affect the lens. We'll talk about that today. That portion of the peripheral nervous system that is not under our control. Okay. Number 3. For number 3, give me the location along the spinal cord for the parasympathetic innervation. What portion of the spinal cord is responsible for putting you in the relaxed mode or the parasympathetic mode? What portion of the spinal cord? There's actually 2 words to that one. Okay. Number 4, in the parasympathetic mode, we find that the ganglia are located either outside the organ itself that it's going to innervate and put into the relaxed mode. They're at the outside of that organ, or they're just within an organ. And this is all the parasympathetics. Where are the ganglia located? We know that the sympathetics had the ganglia in the paravertebral or the lateral. Where do the ganglia exist in the parasympathetic mode? And that is for number 4. Okay. Those 4 referred to what we discussed several days ago. Now, let's go back to our diagram and continue discussing structure and function of the eye. We talked about the canal of Schlemm absorbing excess fluid, the aqueous humor. It's constantly being made by the ciliary body and absorbed by the canal of Schlemm. Now, sometimes this canal of Schlemm will fill up, or something will occlude it so that it doesn't drain correctly, or the ciliary body can manufacture too much of the aqueous humor. In this case, if there's too much of the aqueous humor here in the anterior chamber, it will put a great deal of pressure on the lens, which can put pressure on the vitreous body, forcing that vitreous body to push against the retina very tightly. If it pushes against the retina very tightly, it can actually cut off the blood supply of the choroid. It can push it so that blood isn't going through the capillaries of the choroid. And if blood doesn't reach the retina through that choroid coat, then the retina will start to die. This is what we refer to as glaucoma. Too much pressure in this anterior chamber, and the pressure is building up because the fluid is not draining through the canal of Schlemm, or because there's more fluid than is necessary going into the anterior chamber, putting undue pressure on that retina, and undue pressure on the choroid, cutting off the blood supply to the retina, and the rods and cones will start to die. When that occurs, it's really, glaucoma is an insidious disease, and it usually starts in the peripheral vision, and it is so slow and insidious that it closes in. But when it gets to this area, right in through here, you start noticing that it's black. You've lost your vision. And that's when you may go to the ophthalmologist or the optometrist, and they can do a little instrument. They can place an instrument on the cornea itself and measure the density or the pressure in that anterior chamber. Or you can put your little chin in a depression, and the ophthalmologist will blow a poof of air. And when he does that, he measures, or she measures, the intensity of the waves made by that aqueous humor in response to that puff of air. And that's how they can decide whether or not the pressure is increasing and that glaucoma can occur. By the way, there are 9 different types of glaucoma. Many of them can be corrected by simple eye drops or surgery, but some cannot be corrected, and some forms of glaucoma can lead to total blindness. Okay, so we've talked about an astigmatism, and we've also talked about the possibility of glaucoma. Now, when light passes from the aqueous humor, it goes through the little hole in the eye that we call the pupil. And that hole is surrounded by the colored portion of your eye, the iris. And the iris is a muscle. Actually, there's 2 muscles. There's one muscle of the iris is circular around the outside of the eye. And if you look at your eye, you can actually see it quite a bit if your iris is light. If it's green or blue or gray or a light brown, you can look around the ring of that iris. And if it's very, very dark, that's the circular muscle of that iris. That's the circular muscle. And then within the inside is the radial muscle of the iris. The color of your eyes, the color of that iris is hereditary. And at birth, most babies have a light gray to a light blue color, which becomes more and more pigmented depending upon heredity. The very best color that you can possibly have for that iris is dark brown, as dark a brown as you possibly can. And the reason for that is that ultraviolet light is damaging to the eyes. And when you have a lot of pigmentation, a lot of melanin in that iris, then when the iris constricts so that the pupil is small in heavy direct sunlight, that pupil will constrict, then you don't get light in through the pupil or too much light through that vitreous body. You want just the right amount. And if you have gray eyes or green eyes or light blue eyes, then ultraviolet light can get through more readily. And that light coming through can affect the biconvex disc. And ultraviolet light over time can cause this convex disc to be cloudy or yellow, and that's when cataracts are identified. And that can be remedied, but it is caused by ultraviolet light. And that's why the most desirable eye color is dark brown. eyes that are almost black that have lots and lots of pigment in them. It's a very desirable color because it's the healthiest eye color you can have. As light passes through the pupil, it then it passes through this biconvex disc that we call the crystalline lens, and light is bent again. In your textbook, there are illustrations showing you how light is bent once it passes through the cornea and the aqueous humor, and bent again as it goes through the crystalline lens. And let me turn the air conditioning on again. It's getting hot. Good. He turns it up, I turn it down. Okay. Now, the beauty of this lens is that it can change shapes so that your vision can accommodate near and far objects. And it is able to change shapes because it has attached on the inside of that lens, and there's an envelope, not inside, but outside. The lens is in an envelope, and these suspensory ligaments are attached to the envelope. And when the ciliary body contracts, then that lens can change shape. It can flatten or become more convex so that your vision can accommodate. And it can do it very, very quickly. You can look across the street, and within a split second, these little suspensory ligaments will tug on that lens, changing its shape so that you can see clearly and also close up. Now, something happens over the age of 40, sometimes in the late 30s. People start to realize, hey, I can't see that newspaper quite as well. And they have to hold it at a far area like that, or they have to, in order to see it, they have to put readers on. That's because one of the things that seems to grow as we grow older, besides our hips, is this biconvex crystalline lens. It will increase in size as we age, and it becomes more unwieldy for the suspensory ligaments and the ciliary body to change its shape quickly, and that this crystalline lens seems to stiffen up and it just can't accommodate. That is referred to as presbyopia, presbyopia, and the common name for that is old eye. Not a very gracious name, is it? But it does occur. And it's okay if you can get some designer glasses so that when you do have presbyopia, you have something of a couture appearance. Okay. Now, light is bent through that crystalline lens, then it passes through the vitreous body. When it passes through the vitreous body, those light rays should fall directly on this area involved with the fovea centralis and a little south of that, just a little downward area. That is the area altogether of most acute vision. Now, sometimes because of heredity, this eyeball may be a little short. It may be shorter than others, or normally, or the eyeball can be too long. In that case, the actual light rays will not fall directly on the fovea centralis. And when the eyeball is a little too long or a little too short, then you can develop myopia or hyperopia, which is far vision, myopia, which is near vision, and you have to have corrective lens, glasses or contact lens to enable that light to refract accordingly and fall on the fovea centralis. The eye can be perfectly healthy. It's just a long eye. Perfectly healthy. It's just a short eye. And that's myopia and presbyopia. Whereas old eyes, myopia and hyperopia. And this one, old eye, is presbyopia. Now, one of the advantages of, if there's an advantage to having cataracts, is they can go in and they can remove that cloudy cataract and slip in another lens. And that lens is not as thick and stiff as the old cataract lens, but they can slip it in a new lens, and it can be to your advantage because it can remedy myopia or hyperopia. It'll be your prescription, and then you can start seeing sometimes 20/20, 20/40. So that's one of the positive things about cataracts, is that possibility is there. Okay. Now, I did want to mention again, the indentation right here is referred to as the fovea centralis, macula lutea, and that area filled up with cones is the fovea centralis. While we're talking about that, I want to mention another condition that seems to afflict the aged, and it can start in the 40s, and that is known as macular degeneration. And there's two types. There is what they call a dry macular degeneration, and when that occurs, debris, which can collect in the eye itself, can collect over those cones in that macula lutea. And if it's over the fovea centralis, then it obstructs the vision and can cause blindness. That is referred to as dry macular degeneration. And the other one is referred to as wet macular degeneration. And that occurs when, for some unknown reason, some of the capillaries that are present throughout the eye, especially in the choroid, they will start to grow over that fovea centralis and obscure the view. They say what happens is you lose the central field, and the only thing you can see is a peripheral vision. So if you take your hands and put them right in front of your eyes like that and look straight on, then looking straight on, you do see, you do have some peripheral vision there. And that's the only thing that's afforded to you. They're treating this with a number of modalities. One of them is vitamins, a certain amount of lutein and vitamin A. They are administered because they say that having an ample amount of these vitamins keeps the macular degeneration from progressing. So there is some hope to that. Okay, now let me mention something else to you. The iris, or the colored portion of our eye, we said that in heavy light, when you go out, say it's a bright sunny day on the beach and you're getting the reflection off that white sand and it is really bright, then your pupils will constrict and they'll get very, very tiny. And that's the work of that iris. And it's caused by the autonomic nervous system. That iris is not striated muscle, it's smooth muscle, and the autonomics control it. So it will cause it to tighten down or constrict in bright light, narrowing that little opening or the pupil. Then when you go into a theater, your autonomic nervous system will move into gear and it will cause your pupils to become very, very large. So that means the iris will pull back, increasing. And if it's dark enough, sometimes that pupil looks like it's taken up the entire eye. You don't see the iris at all. When that occurs, we say that pupil is dilated, dilated, not dilated. We tend to say that in the South, but it's actually dilated, dilated. Okay, I did want to mention that to you. And I'm going to ask you to do, we've been working, good grief, a little while now. So what I'm going to ask you to do is complete the study on that lecture 13 into the area where it's talking about the extrinsic muscles, because you can study that in your textbook. Please do so. And if you look at the nerve supply, lecture 14, I want you to study that. We've mentioned all of those to you already. So this is just a reiteration of what you already know. Now, I did give you 4 questions on your quiz. Let me give you number 5. Number 5 pertains to our discussion of the eye. And remember, I just mentioned to you a few minutes ago that when you walk into a dark theater, that your eye has to accommodate that. We do know that the cones fill up, not the cones, but the rods fill up with a rhodopsin so that you can begin to pick up on light, and that the pupil enlarges a great deal so that more light does come in. And what is the term we use for enlarging that pupil? What is the term we just taught a few minutes ago on the enlargement of that pupil? That is number 5. Okay. Number 6. In number 6, you can turn to your textbook and look at this in the reflex arc, or you may already know it. Hopefully you do know it. In the reflex arc, when a sensory afferent neuron picks up, say, on pain in the body, and those afferent sensory neurons are carrying the message to the spinal cord. In the reflex arc, we do know that the sensory root goes to the back side of the cord. What is that ganglion called in the reflex arc that is right next to that dorsal root? What is that ganglion called that is involved in the reflex arc on the sensory side? That's number 6. Number 7, what is the name? Give me the name of those special photoreceptor cells in the fovea centralis that picks up on color. The photoreceptor cells in the fovea centralis that pick up on color. Okay. Number 8. Number 8. This is a downward projection of the sclera. It's frequently referred to as the window of the eye, and it is convex. It is convex in structure. What is it called for number 8? Number 9. Give me the name of the photoreceptor cells in the retina, which are responsible for picking up on light when the light is very, very dim. It picks up on light being bounced off of an object through the refractive media of your eye, and it does so in very dim light, and it uses that rhodopsin pigment. What kind of photoreceptor cell is that? And number 10. Now, number 10, you'll have to read the next lecture. It's just a very, very short lecture. I want you to give me the name of the cranial nerve that has 3 branches on either side. And one of these 3 branches from this cranial nerve picks up on pain in the ophthalmic region. What is the name of that? It picks up on pain in the ophthalmic region. What is that nerve called for number 10? Okay. Now, what I would like, let's see how many minutes we've been working. What I would like to mention to you is that this weekend, I'm going to email you your list of terms for your final exam. And your final exam is going to cover the 16 units in neurology and just 2 of the units or 2 of the lectures in the dermatology. That'll be 18. That's plenty of material to work on. But I'm going to send you the terms. I had originally decided to pick out 50 words, but 50 words didn't do it justice. There'll be 60 terms that you'll have to know, and they're all terms that we have discussed in class or terms that we have discussed online in your lectures. And so you'll have all week, a good long time to study each one of these terms. What I'm going to do on May 7th at 9:30 in the morning, that's the time that Southwest says we can have for our final. Then what I'll do is I'll choose 33 or 50, I haven't decided yet. Of those 60 terms, I will give you the definition and you'll fill in the blank. And what I'll do is you'll number your paper 1 through 33 or 1 through 50. And as I call out the question, then you write it down for number 1, the answer to number 2, the answer to number 3. And when the test is over, then I'll give you time to email me that information back. And you can use a word bank in this. I've never done that before in my life, given any student a word bank, but you can have it. But it would be difficult if you didn't know the word. So you really have to know these words well. You'll have the word bank there, but it's not going to be that helpful to you if you don't know these words, know their spelling. So I want you to do that, and I'll give you ample time. If I give you 50 questions, then it would be 50 minutes. I'll give you 50 minutes from the end of the exam to email me. What you'll do is you'll number your paper when you email me, just like you've been doing on these quizzes. Instead of 1 through 10, it would be 1 through 33 or 1 through 50. And you'll type these in, and I'll give you plenty of time to do that. Okay, if you have any questions, please call me. I will try to get this word list out to you. And if not, if I don't hear from you, I'll get the word list out to you, and then I will work on lecture 15 and possibly 16 the next time we meet next week. So please call me 761-3636 if you have any questions. I sure miss you guys. I enjoy teaching you in class so much more than teaching a plastic camera. Okay, God love you, and for sticking with me. Stay healthy, please.